So now we move on to the DDL annual lecture. And I'm very pleased, delighted that this year we have Professor Hugh Smyth giving this lecture. And I'll give a brief introduction to Hugh, because I think you really want to hear from you and not from me. There's a lot of things I could read out, so I'm going to give a very brief capture here. So Professor Smyth is the Alcon Centennial Professor, College of Pharmacy, the University of Texas, Austin. Dr. Smyth oversees a multidisciplinary laboratory funded through the NIH, FDA, and industry that focuses on engineering new drug delivery systems with an emphasis on drug delivery to the airways. He has over 300 published works, of which have been cited over 6,000 times. Dr. Smyth has also started several companies resulting from technologies discovered in his laboratory, including Respira Therapeutics, Nobhill Therapeutics, Via Therapeutics, and there are more. I'm going to stop there because I really, really want to hear what you have to say today, Hugh. Thank you.
Thanks very much, Gary, for the introduction. Is my microphone working?
Yep.
It is. Okay, great. I'm very honored to be selected to give the DDL annual lecture. I was very surprised when I got the email from Gary. And in putting together this talk, I wanted to kind of give an overview of the field itself, but with some of our own personal research that we've been doing to address this. What have we learned? Where have we come? And where are we going in the field of drug delivery to the lung? I have a few disclosures, but I guess I'll start with a survey I actually did informally about 5 years ago for an IPAC ISAM presentation. What I did is sent a bunch of emails to some key opinion leaders in the respiratory area and asked them what was the most important advance in our field that they've seen in the past 10 years. And I got a lot of different responses. And to me, what this was indicating is that we've been, as a field, been working in many different areas and many different problems, solving many really important problems that we've been having in drug delivery to the lung. And you can see here there's a list of maybe 10 or 20 different things that people individually thought were the most important advance. And this is about 5 years ago. But I think this list is actually a really good list to sort of inventory where we've been and what we've learned. Along the way, though, I guess with the research that this field has been doing, we've discovered that there are other barriers that are going to need to be overcome to deliver the next generation of therapeutics to the lung. These include, the physico-anatomical barriers of basically delivering aerosols to the lung. And I'm going to talk about one case study that sort of encapsulates our fixation with aerosol efficiency and performance. But there are other barriers that are really important for the next generation of therapeutics, including extracellular barriers, things like mucus, cellular barriers, how do you get your therapeutic cargo into the cell, and then more molecular barriers like stabilizing biologics. So just to summarize what I'm going to talk about, I'm going to look at essentially what our focus on increased aerosol efficiency has given to the field. Essentially, we've been trying to improve aerosol performance, but along the way, we've actually learned a lot more than just how to improve fine particle fraction. The next part I want to talk about also is biologics, because I think that's where we're going. And we've actually been a long way with biologics, have a long history in the field. So what do these successes and failures mean for us? And then I'm going to have a series of vignettes of where we're going as well, looking at these extracellular barriers, new types of therapeutics, looking at different modalities of administration, and even processing and manufacturing. So one of the really important papers that I get my students to look at is this paper from 1971 by Bell and colleagues, which I think was a landmark paper in our field, which talked about dry powder aerosols. This was essentially the technology behind the spinhaler. And if you go back to this paper, you will see that what we talk about today was actually what the very first paper and science paper for dry powder inhalers was talking about: carrier particle size, aerosol performance, emission of the powder from the device. Really, really interesting paper. But what that tells us today is, the basic design for dry powder inhalers hasn't really changed. Yes, I'll talk about some major innovations, but the basic design has remained the same, where you have carrier particles and fine micronized drug particles combined together, and we put them in a device and they're inhaled. But what we also have known since 50 years ago in 1971 is that these fine particles are very cohesive. And our design window, the 1 to 5 or 1 to 3 micron size window, means that we don't operate in the gravitational physical world. It's more under the influence of van der Waals forces. And so as a result of that, we generally have low efficiency of formulations and dry powder inhaler devices. What's the consequence of this? Well, it is important. It would be good to get more API delivered to the target tissue. But importantly, we'd also have, as a result of poor performance, we have flow rate dependency. And so low performance and flow rate dependency is obviously not good for the patient. So I think that this has led to, at least in my group, we've been trying to figure out how to improve performance, improve efficiency. What are the factors that influence manufacturing and design of these types of systems? But essentially, it's about changing this balance between aggregation or particle adhesion and dispersion. And so one of the things I think the field, as well as my group, have been focused on is, again, this concept of fine particle fraction, respirable fraction, or improving efficiency of aerosols. And when we started looking at this maybe 10, 15 years ago, we looked at carrier particle size as one of these parameters. And this is just an example of one of the studies that we're interested in. But if you went back around, I guess, 15 years ago, 15 to 18 years ago, you would see that what the literature was telling us is that as you increase the carrier particle size, you decrease aerosol performance from dry powder inhalers. However, if you looked at how those studies were done, they were done in sort of isolated systems where some morphologies were looked at across different size ranges, or across a single size range, multiple morphologies were looked at. So what this dogma in the field of that increasing carrier particle size would lead to decreased performance was based on sort of an incomplete data set. So what we endeavored to do was to do a systematic study. And so what we did was, what one of my graduate students did was to essentially make 13 different size fractions from commercial grade lactose. And these were triple sieve to make them very, very narrow particle sizes in each of these size range buckets. What he did was then look at 4 different morphologies of commercial grade lactose. And he also tested this across 2 different drug classes. So there's about, I don't know, 50, I think 56, maybe more individual formulations that were tested across many different replicates. And essentially what he found in many cases was what the literature was always telling us, which is as you increase the carrier particle size, you decrease the respirable fraction coming out of these dry powder inhalers. However, in certain cases, what he found was that depending on the morphology, you could see increases in aerosol performance as you increase carrier particle size. So we started to investigate that a little bit more. And what this led us to was one of our, I guess, most important findings in this field was that depending on the morphology of your carrier and depending on the device design on how it essentially interacts with the formulation, you could shift the detachment mechanism from being a sort of aerodynamic shear detachment from more of a momentum change or a detachment by impaction. And we spent a long time sort of trying to validate that. So what did we learn? We learned that significant detachment forces can arise from particle collisions. And then we began to think, well, could we utilize this to improve aerosol efficiency? And so this was the aha moment for my graduate student when he used some simplified assumptions for how particle adhesion versus particle detachment would be as a function of carrier particle size. And essentially what he saw is that in the previously studied regions, there's this very equal balance between adhesion and detachment, inhibiting aerosol performance. But if one were to increase the carrier particle size well beyond sizes that were currently used in dry powder inhalers, you could potentially get much greater forces of detachment compared to the adhesive force. And so after another couple of years of work, we came up with this system where we could use a single carrier particle, a single sphere, a bead of about 5 millimeters, 3 to 5 millimeters, coat the drug onto the surface of the sphere, and use a specific chamber where the drug-coated sphere was in, and essentially we could get really high fine particle fractions based on this collision or momentum transfer that was very efficient at dispersing fine micronized drugs. And so we tested this across many different doses of drug, many different types of APIs, and showed that, yes, detachment forces can be significantly enhanced by momentum transfer. So, as I mentioned before, the field has been focused on fine particle fraction and aerosol performance. And over the last 20 years, there's been many really important technologies developed that will achieve high fine particle fractions for certain APIs. So we've got the low density particles, the ability now to modify particle shape with this exquisite control. We've got commercial products with magnesium stearate that not only promote aerosol performance, but protect APIs. We've got a huge array of device technologies now that the field can turn to if they need a device technology to match up with a formulation or an API. So I think what we've learned is that there's many different ways of achieving high fine particle fractions. And I guess Dr. Lavarini, in her opening presentation, showed a very similar slide to this one, where you have the increase in fine particle fraction over the year. And so basically, as we've been as a field working on this problem, we've shifted the needle and improved things substantially. But I think it's not just efficiency that is important. I think what has resulted from these technologies, these formulations, these devices, is a great benefit to the patient by things like reducing inhalation dependency of our systems. And so Toby Podhaler is one example where you can see that there is low dependency upon how the patient uses the device on what dose they're getting to the target tissue. And then again, referring back to the opening plenary lecture by Dr. Lavarini, it doesn't matter how efficient our systems are, our formulations or devices, if the patient doesn't use them. And so, important advancements in the field have been related to that: the open, inhale, close, the 3-step type device, and also the connected inhalers, which I think, the jury is still out on whether these are really going to improve outcomes of patients. So where are we going? One of the areas that I think the field is moving towards is different therapeutic opportunities. And these are, not using potent molecules that you would see for asthma and COPD, but these typically are low potency opportunities where high doses must be delivered. And actually, we've been here before. For example, the Aerohaler, the Abbott powder inhaler shown up here in the figure, included 60 milligrams per dose. The Spinhaler, 1971, referring back to that, was also a relatively high dose, 20 milligrams of API. Obviously, the efficiency of those systems didn't mean that high doses were actually delivered to the lung. So I think there's a number of devices that have been described in recent years. The Toby Podhaler. Here we have 28 milligrams of powder that's in a capsule. 4 capsules are inhaled. Orbital, Twinsa, TwinMax, all devices designed to meet these new needs of low potency molecules. And so we've been working in this area too. And one of the APIs that we've been particularly interested in is clofazimine, which is an old antibiotic that was used for the treatment of leprosy. Now, it's recently been found to be highly effective against mycobacterial infections of the lung, like NTM and TB. In this case, we're just using a off-the-shelf RSO1 with 20 milligrams of pure drug powder in there. And really surprisingly, what we see is high emitted fractions, high fine particle fractions, regardless of the pressure drop. So a 1 kilopascal pressure drop versus a 4 kilopascal pressure drop. With a high pure drug, no excipient powder, we can get 20 milligrams of this API delivered. Another technology that I'm involved with is Nob Hill Therapeutics, which has been developing a dry powder nebulizer, where essentially an external energy source is used to create a powder aerosol within the device from which the patient will inhale using tidal breathing to deliver high doses of powder. And there's a poster downstairs, poster 80, that describes this technology. Where are we going? Well, with biologics, we've learned a lot, but I think with new therapeutics, biologics are going to be leading the way in inhalation therapeutics. The pipelines for biologics are rich, not just for inhalation suitable molecules, but just generally. And I think there's more than 300 of these in development pipelines. And oftentimes we're asked, well, haven't we, sort of seen this before and, it didn't work. And I think, the Exuber experience from an outsider's point of view, makes people very skeptical about inhaled biologics. But I think most people in this room would agree that there were many successes with inhaled insulin, not obviously exemplified by these blog pictures that people have created about the Exuber inhaler. The reality is that we've actually been delivering biologics for over 30 years. Pulmozyme, for example, was approved in 1993. We do have an inhaled insulin on the market. And there's, if you go to clinicaltrials.gov, for example, there's hundreds of studies that have been initiated looking at peptides, proteins, and other types of biological systems. And I guess another informal survey that I have been doing is asking our colleagues in the, I guess, preclinical area of what type of programs they're seeing. So in preclinical toxicology programs, development programs, what I'm hearing is that about 50% of these programs are biologics now, as opposed to small molecules. And these are very diverse biologics. They include gene therapies, bacteriophages, siRNA, CRISPR-Cas systems, things of that nature. And I think this is where we're going with gene editing, gene delivery, mRNA technologies. And what we have learned, actually, if I just go back to this slide, is that making these formulations and delivering them is not that hard. We can spray dry monoclonal antibodies, and they're very stable. They're not degraded if you formulate them and design the spray drying processing parameters correctly. However, I think what has also been revealed about our experience in gene therapy over the last 20 years is that there's many barriers still to be overcome. And these are more of the extracellular and cellular barriers that I talked about at the start. And these are ubiquitous to the delivery of nucleic acids as a therapeutic. One of the most important ones that we're going to face in the airways is these extracellular barriers like mucus and bacterial biofilms. So mucus has been described as a combinatorial binding library, and it is one of the main reasons why many of the early gene therapy programs that were inhaled received low efficiency or low transfection rates in patients. It's been widely shown that these early vectors for gene therapy are very susceptible to binding to mucus. So there are ways of overcoming this, though. In our lab, we've been looking at chemistries to overcome mucus binding. Here's an example of a small molecule, tobramycin, that we pegylate that allows it to get through both mucus, cystic fibrosis like mucus, and also Pseudomonas biofilms, and then kill the bacteria. We've also applied this to polymeric gene delivery systems and to lipid nanoparticle-based systems. One of the other major barriers is going to be overcoming the physical stability of carriers like lipid nanoparticles, which are so promising. Here in this study, we looked at a wide range of different lipid nanoparticle formulations and found most of them were very unstable upon vibrating mesh nebulizer aerosolization. However, we found a few that were resilient to those forces and could, in vivo, also achieve high transfection efficiency. Another thing that we've learned relatively recently is that even commercial products with advanced manufacturing have batch-to-batch variability. this leads to significant consequences for the patient, different pharmacokinetics, and also for generic manufacturers who are trying to match up their devices and formulations to the RLD. And so we've started studying this with help from the FDA, looking at batch-to-batch variability, both in terms of aerosol performance and dissolution of the API. And it's too early to give the conclusions of this study, but essentially what we're finding is there is very subtle physicochemical differences between powders in these batches that can be detected. And this will be very interesting moving forward in terms of both regulatory, but also designing new formulations with low variability. So where are we going with manufacturing? Well, another thing that we've been interested in is continuous processing or continuous manufacturing. And in collaboration with colleagues who use twin screw extruders, we've been using this as a twin screw mixer and starting to develop a mill to fill continuous process where the mixing of these carrier micronized drug particles can be achieved in a continuous manner using these twin screw mixers. Finally, I just want to end on this last slide where I tried to sort of update my poll that I showed you at the start by doing a LinkedIn poll. I only got 69 responses and not too many comments. But I asked, what is the most impactful advance going to be in the next, I guess, 7 years? And surprisingly, people are still thinking devices and formulations are going to be very impactful. Obviously, new therapeutics will be impactful to patients. But I would like to acknowledge my group. Many of the people on this slide contributed significantly to the papers over the years and to our understanding that we've developed. This is some of my current group. I'll be willing to take any questions I see. I've just finished my time.
Thank you very much. It was interesting. I think a lot of the things you talked about there, Hugh, probably resonated with a lot of people in the audience. Do we have any questions? So put your hand up. We can bring a microphone to you or go to the standing mic, or again, feel free to use Slido.
Perhaps I'll start while people are thinking and making their way. This has come through from Slido that I think it reflects on, you mentioned this, so at the start, Professor Lavarini said one of the issues that we face is healthcare providers and their lack of understanding about inhalation technology and how it works. Do you think some of these developments, perhaps the new therapies or the new process technologies, is going to make that, add to that problem, or do you think it will start to address that problem?
I think if our new technologies are less sensitive to patient use or patient variability, intrapatient and interpatient, then I think that just makes the physician's role a lot easier. It makes it easier for them to prescribe a system that they know is going to be more predictable and working for their patients, multiple patients. They might not need to select a particular device given the idiosyncrasies of their particular patient that they're seeing at that particular time. So perhaps I think these technologies will make it more seamless for physicians. I think we're a long way away from reaching that goal though.
Unfortunately, yes.
Thank you. I think we've got time probably for one more question. Have we got somebody over there?
Hi, Murat from Vectura. Actually, it was a question around patient variability, and I just sort of wondered how much of a factor that is in, the performance of, of the drugs, and how much we should be looking into that more.
Yeah, really great question. When we do our in vitro testing in the lab, right, we're testing at sort of typically constant flow rates, constant volumes. But if you were to compare the variability due to the patient and compare the variability due to the device or the formulation, the patient is going to be many orders of magnitude more variable. There's been many studies showing that inhalation flow rates in the morning are different within the same patient than inhalation flow rates and respiratory parameters in the evening. And so the variability in the patient is the largest variable in the room. And so that's, how we should be thinking about designing our technologies and our systems to accommodate large variability. Testing at using realistic inhalation profiles, but at the extremes of those to test our formulations, our devices, to show that despite variability within the patient, we can achieve consistency in dose delivery.
Okay, thank you. So it's my great pleasure to award Hugh a rather splendid gift. You can't see this, you probably won't, but the picture will be on the website later, so please have a look.
Thanks, Gary. Thank you.